started. Thank you all for coming. It's my great pleasure to introduce our today's speaker, Eric Xing. So Eric currently is the president of the Mohamed Bin Zayed University of Artificial Intelligence in UAE, and he's also a professor here in the School of Computer Science, spanning several different departments, MLD, LTI, and CST. And Eric has been really a pioneering leader in developing, not only machine learning methodologies, but also many applications, and not only the methods for ML, but how to run these methods at scale. So his research has been, a wide range of important topics in both the fundamental methodologies and also their applications. And in particular, recently he has been working on, foundation models, world models for important applications in health and biology. And I think today we're going to hear a lot about his recent work and, very importantly, his perspective, because I think this is a topic that is of great interest to many of us here and also certainly the broader community. So Eric, thank you very much. Look forward to your talk.
Great. Thank you, Jian, for the very kind introduction. Yeah, so I'm going to talk about toward an AI-driven digital organism. It is a multi-scale foundation model vision for predicting, simulating, and programming biology at all levels. It's kind of maybe a report card back to CMU about what I did in the past few years in Abu Dhabi. Last Monday, I talked about a similar grand vision for word models and action models after the LLMs. And this one is the other branch, how to use a similar mindset, I would say, or maybe philosophy to solve biology. Not the same method, but the same philosophy. The details of today's talk and the vision, the position are summarized in this paper, which was supposed to be out last week, but Thanksgiving slowed down everything. It's still in somebody's desk in archive. So hopefully this week it'll be out. It's a 30-page paper, lay out maybe 30-year worth of work down the road, to solve biology. I welcome comments and suggestions. Now, just to position us, on the right footing, I think we all see this curve many times in various different forms. Data-driven approach, data-driven, data-enabled, large language models, foundation models are really booming, and they are now everywhere, in the space of human cognitive activities, linguistic activities, visuals, and so forth. And there has been debates, you know that, right? So are these models really intelligent or not? They are super smart in solving, math, IQ, standard tests, and so forth, beating people, in many different challenges, right? But there are people also argue that they are not smart. They're not even as smart as a dog because they don't do so on and so forth, right? This kind of conversation has been on and on there for a while. My take is that it is a very boring and useless conversation because we build tools not to make it smart. We build tools to solve problems. You build an aircraft. It's not because you want to fly with your wing. You want to go somewhere through aviation, right? So if you try to frame your question and your task in a utilitarian type of mindset, suddenly, whether it's intelligent or not, that problem is irrelevant. It's gone. Right? And whether, for example, this machine understands knowledge, whether it generates knowledge may not be relevant if that's not what you want. If what you want is an answer, then you take the answer. That's it. That's the very, very simplistic and brutally honest attitude. Now, along this line, you may be asking for the following, right? What else we can do if we have a model that can predict or solve your problems? What may be your other problems? So here I have a few other problems. Maybe you want to simulate human cognitive and behavioral activities and the outcomes, outside of linguistic communications. Right? Maybe you want to simulate a physical world, how water flow, how weather, evolve, and so forth. Maybe you want to simulate social behaviors so that you can do econometrics, with simulations. So there can be models built for that. For example, nowadays, people are building the word model to simulate the physical world, or even the human behavior activities and the agents and so forth. And not necessarily even knowing the mental states and the law of physics, but as long as they can simulate, why not, right? So in this topic, this particular talk, we are going to talk about simulating biological activities at all levels. Why? Well, obviously, it's interesting. You want to apply a drug to a cell. You want to see what's the outcome. That's a simulational problem. You want to explain the outcome. That's a different problem. In fact, when medical doctors give you a pill, give you a solution, I'm not even sure they notice the explanations. They just give you a pill because that's written in the medical book. Right? So it's a very, very acceptable approach if you just put down your ego and just want to consume and enjoy the results. And why, in particular, biology may enjoy a simulational system? Well, there are additional reasons about the utility. Because biological systems are very complex. In fact, part of the journey that we got trained is to use the first principle to do everything, right? So for example, in biology, Schrödinger has been advocating using physical and chemical laws to explain biology. To some degree, he was successful. He was able to predict that the genetic codon was harbored in some aperiodic crystal structures and so forth, which gave rise to physicists studying DNA crystallography and so forth. So there's some success. Max Brück, Crick, Watson, Pauling, all follow the same spirit and push more a mathematical and a physical law-oriented approach in understanding biology. And we do have a dream that we may be able to understand biology like we understand the celestial system using Newton's laws and so forth. And we can predict, life, just like we predicted the position of a planet and so forth. That's the dream, right? That's actually what is the holy grail of a first principle driven approach. First principle approach was successful to some degree in biology. For example, the Mendelian law of genetics, of inheritance, genetic codon, coalescence, to define the bottleneck population during evolution. Yeah, there are some good theories behind it, and we know exactly what's going on. But there are more failures. For example, we don't have a model for the phenotype. Okay, we don't have a model for cellular dynamics, disease model. You have a flu even. We actually don't have a system that can reproduce a flu on another person, on simulators and so forth. Right? So biology is just too complex to compute based on first principle. I think that many people realize that. Now, the question is that, do you wait for the math to evolve in a certain way so that you can apply first principle to compute biology, or you will go through some other approaches? It's very difficult in first principle. I think I will give you a few examples. For example, to model biology with math, you need to face the first challenge of biological systems speak a different language. They don't actually, speak the same language as we did, if we do in human language. And they use central dogma, and they use, nucleotides, to encode a lot of informations. There is a process of cascading the information from DNA to RNA to protein, and then coming back, to influence the behavior of the cell in a closed loop. Not only the language they spoke is different and also very, very multimodal, their scale is also massive. We're talking about phenomena as small as a nanometer scale to the physical scale like a meter, like a human being. This whole spectrum is actually in action when you want to ask a single biological question. For example, I ask you, why do you get a flu? Okay, you're starting from here. You can go all the way down to what's going on in your cell. And there may be, a defect in your DNA, not for flu, but for other diseases and so forth. Therefore, the solution or the answer or the remedy has to basically cover this entire spectrum of problems. And also there are spatial temporal issues, like this embryogenesis, of animals. The cell basically undergo a journey, depending on where and when, it is situated, they will behave differently, even though they all have the same gene, same DNA, but somehow it was programmed, but also conditioned on the environment so that the behavior of the cell can be different. And then there are also sudden emergent behaviors. In this case, it's not a emergent for the good. It's actually emergent for the demise. That in the Alzheimer patient's brain, in fact, at a certain time during your age, all of a sudden, the brain structure gets changed and the neuron cells start to shrink. People believe that it is encoded in the gene already in your genome, but people don't know why it is happening at age 70 or 80 or something. We don't actually know. Right? And some people actually don't have this kind of problem at all. We don't know either. So that brings us to maybe the opening part of this talk, which approach we want to use to understand biology even before we know all the details in mathematics. The Kantianism, is really the foundation behind first principle. In fact, all of us, the modern science was greatly shaped by Kant, his philosophy, that we want to use rationalism and reductionism, and we also can use the prior knowledge as a form of knowledge, without even evidence, to drive the inference of knowledge, and then to understand and deconvolve and decompose the anatomy, and the mystery, in complex phenomena. And you can imagine trying that for biology will be very hard. The other school of philosophy, which was kind of condemned in physics or in science, I heard a new article saying that Hume basically destroyed modern science if he was not countered by Kant. His approach is that we should believe in nothing but data. Knowledge only exists in data, and therefore, your prior knowledge, for example, your prior understanding of 1 + 1 = 2 shouldn't be called a knowledge. It's actually a summary of the empirical evidences. That's his kind of approach. Therefore, it is totally fine to pursue, a black box prediction as long as you have actionable empirical understanding that can be repeated and can be robust. The discovery, for example, of vaccine is a prime example of the success of empiricism in biology. When Pasteur, applied some, less active biomaterials, derived from, antigen, into some animals, and he found that, these animals become immune to that disease. And remember, at that time, there is no knowledge about protein, no knowledge about DNA, no knowledge about genetics. But people actually believe in that repetitive evidence and then applied the vaccine, which saved a lot of people. So you don't have to know basically the why, but you can still have faith on the results. In some sense, GPT and transformers is a kind of a empirical approach. We don't actually have a system that actually understands the linguistic law and the rules, of our, vocal capability, communication capability. But this whole system is trained on large amount of linguistic data, so large that they include all the data ever created in civilization. Basically, all the answers that you could have generated in the literature are somehow encoded and ingested, in such a system. And there are also these engineering tricks to make them even more adaptive and more aligned and so forth. But at the end of the day, what you see is the other end of GPT. You see an agent which can answer almost all your questions better than any individual in the world, right? Okay, do you believe in or do you call it intelligent or not? No, it doesn't matter. It's a utility that you may even feel safe to use these days. Depending on what's your criteria of safety and also what's the nature of utility. For example, I see many very important senior people using the GPT to draft their speech, and then spend another 1 or 2 minutes to polish, and that saves them in a few days of work from their senior aides and so forth. I'm talking about very senior people, rulers of the countries and so forth. Okay, so they feel safe. And in fact, similar success already are seen in biology. We've seen, a transformer-like architecture, being very successful, in structural biology, making predictions of protein structure, through the system called AlphaFold. In fact, recently, the award of a Nobel Prize in chemistry to this kind of, underscored the significance of this breakthrough and also the validity of this solution as a tool to advance science. So we ask, can we do even further than that? because the biology, is very, very complex. It's not just a protein. In fact, biology tasks oftentimes are across the scale. I mentioned about, for example, understanding disease require you to basically penetrate through the layers of biological granularity to understand cellular behavior, molecular behavior, and so on, right? But if you have other tasks like drug discovery, target discovery, drug design, or clinical diagnosis, in fact, even if your problem is at the scale of this end, you still need to have knowledge about that. Not knowledge, but have a capability of simulating that. Because if you design a drug, what's next? What's your trial? You need to basically know if the drug is interacting with your target, what's the function and what's the structure. What if the solution in the cellular environment is unfavorable? What's the melting temperature? All these actually require you to have a holistic system that allow you to at least either simulate, or experiment, in real life with your predictions and designs. And that's why we, need a more holistic approach to medicine and to biology. And that's what the current science is offering. For example, I think in the world of methodological domain, we are still in the stage of so-called one disease, one organ, one cell type per model approach, right? It's always a uniquely designed specific approach. It's almost like becoming a doctrine, of our research that you should focus on this, and therefore your model does only this. That's how we were trained. In fact, if you look at the departments in biology, they are called biochemistry, cell biology, microbiology, genetics, and so forth. Each of them is actually study a slice of the phenomenon of the same thing. You may be actually breaking down a bacteria and take the molecule out to go to biochemistry, and then take the sequence to genetics, and then take the cell to the cell biology. It's the same cell. You need to divide that in that way. Why? I don't know. Maybe it's because it's too complex. We need to divide them so that people, can study different aspects. But it's not the best way to offer a holistic solution like I was. So in recent days, I think inspired by the success of transformers and the LLM and so forth, we start to see emergence of so-called foundation models in biology. Here is a chart, talking, showing you, a short list of such models ranked by size and also, different data modality. I want to make a small comment in here. I think this word foundation model has been abused. Okay, foundation, not transformer doesn't equal to foundation model. Transformer can be tiny. They are not really foundational. In my opinion, to make something foundational, you need to really be big enough, and you need to actually have multiple capabilities. So in here, I just pinpoint a few, issues that I personally had, and many of our colleagues in the community had with these models. They're small, they're highly specialized. They focus on one or two modalities, right? And they actually just borrow the LLM as it is, and then turn the DNA language, the sequence into, a natural language with the 4 tokens, and then you start to train. And you call it the foundation of biology. It's bizarre to say that way because it is, a model not even meant for biology, but now we turn the biological data into a natural language data. Whether it's working or not, we don't actually know. I think here are some of my personal take about how to work with the foundation model approaches. First of all, you don't call everything foundation model just because it is transformer. Okay, you make it foundational. It needs to be very big, and it really needs to reach a certain scale, both in terms of a parameter size, data size, and complexity, so that you start to see the scaling law at work. Okay, you need to climb the scaling law to be at a certain height so that it is actually ingesting knowledge from data. Otherwise, it's not a foundation model. You can easily either overfitting or underfitting the data. And secondly, a foundation model is by definition a generalist. It is not a specialist. What is the LLM? LLM is not made to be a chatbot. Okay, it is made to do next token prediction. Okay, you have all the functions of chatbot, QA, sentiment, and everything embedding from that model, but that model is not made for any of this one task. Okay, they were made for a very, very intrinsic, training loss, which is a MLM or CLM, something like that. And then, of course, the LM architecture are inadequate for biological data, for obvious reason, because biological language is different from human language. And therefore, a new architecture probably is needed. So with that preparation, I want to put a question out there now. We have now, a wide spectrum of biological tasks, all the way starting from molecular design to, public health, food security, agriculture, all dealing with one way or the other a biological issues underneath, the subjects. What if, and also we have all the data. We have data of a huge variety, a gene bank, a PDB, for protein, and nowadays, we have the human protein atlas, human cell atlas, and the phenotype projects. So we are really in the world of multimodal biological data. You can actually look at a biological entity from many different, aspects and the scale and also length. And they can all be used to answer every of the question above. So what's missing here is a holistic and integrative system, which really can be receptive to different type of data and also different type of biological mechanisms, and maybe predictive problems. And what if you have such a model? Then you can, either treat it as a BioGPT, you do inference, meaning that you throw in a prompt and you get maybe some nice prediction classification of cell types, regression to cell melting and protein melting temperatures and so forth. You can also start to fine tune if you have a really good labeled supervised data. You can actually specialize that model based on the pre-trained mechanism, which give you good representations, for example. You can also start to use the model as a generative system to predict or to generate scenarios. For example, what if I CRISPR two genes out of your a whole system, what the morphology of the cell will look like, and so forth. You can start to design reversely. If I like this particular protein structure that binds a cell, what the sequence should look like. And you can maybe even explain by systematically probing the biological systems. So such a system is what I believe to be needed for the next generation biological research, which is a departure from the one model, one problem, and one data approach. And this is happening at least now in natural language processing. We have one model. You probably see that all the past NLP researchers, parsing, part of six tagging, and the topic models, they are all gone because you have now a platform level solution, and then your maybe hope is to lock onto this new platform and then do the next level of work on top of that. I think biology are not yet at that stage, but I think it will happen. It's about really what it takes to make this faster and more rewarding. So we're talking about building such a complex system, right? And people, engineers are very good at doing that. Scientists usually don't worry about system building, but I think in this case, the engineering needs and the awareness is very, very important. For example, in building a plane, what do you build? What do you build? You break them into modules, right? And you build parts from different factories, different teams. But on the other hand, you build them in a very reusable way so that they can be assembled in a very easy fashion. They talk to each other, they harmonize, and then they start to function as a whole. It's like training an orchestra. This is not a very, very appreciated practice in academia, but I think as biology, working out of the closet of medieval workshops and entering industrial age, this kind of transformation is probably needed. And in that case, here is a vision, that I want to share. In the past several years, I've been busy, working in UAE and also in CMU. People may say that, yeah, Eric was making the university over there. Yes, I did that. But in fact, other things I did is to really broaden the scope of the academic program and do some bigger science that is only possible with a university-like infrastructure and also, the manpower. So this is a vision which we called the Jian multiscale model for biology. Jian is the name of that project. The goal is to create such a system, which we will call it an AI-driven digital organism, which contain all these different modules of submodels, but they come together to form the bigger model and hopefully fulfill the problem of the task of predicting, simulating, and programming biology, but at all levels, not just at one single level. And I think to build such a system, such a vision, really requires some sophisticated and also a very well-planned engineering roadmap. And here I just give you a quick kind of a review about the roadmap we are taking. We've been right now working very, very actively on stage 1, which I call divide and conquer. Basically, you just take all these models out separately and train model on each data modality or each field data modality. For example, you can imagine training something on DNA to make it into a DNA foundation model, on RNA and so forth. So you may say, yeah, this is what everybody is doing already. Nothing new, right? So we've seen many models we just say already. What's next? Well, there is a tremendous opportunity remain to be unlocked, which is to really understand, first of all, how to make the model powerful enough, make the best use of data with the right design and the size of the model. That question isn't answered yet. People basically do a one-off experiment. I ran into colleagues who say, hey, Eric, I've got a great model, which does this and that. And I said, how many machines do you use? They had 2 machines. Well, then I cast a doubt. You use 2 machines. What if I use 200 machines? And do you think there's a difference or not? I will show you there are differences. Okay. And also, does the model plug to other models? Right? It's a standing alone model for only DNA or RNA, or you expect that to be part of a system. So having the system view ahead of time is useful in designing models which has the right interface. So that's kind of the opportunity, even in stage 1, I want to share. For example, we need to have some justification about maybe whether the model is having the right size or not. With my colleague Lesson and his team, we did a very, very exhaustive scaling law study in protein language models. And you probably are familiar with these kind of curves, in the LLMs, right? On the upper left corner, you see, each of the saddle curves is basically, defined by a certain amount of compute, the number of flops, which is actually the size of the model times the amount of token that you use. They put together, a defined flop. And you can see that in every given amount of compute, there is always a sweet spot where you get the best size, best performance of the model. Okay, it's not a small size, it's not the biggest possible size, but somewhere in the middle, right? But once you have more and more compute, you can see that there is a straight line that you can actually regress and find that with more compute, with fixed amount of data, your model performs better when it's getting bigger. And we actually don't even know where that curve ends. So people naively say that, oh, with bigger model, I need more data. It's true, but when and how much is an ambiguous answer. And this curve clearly shows that even with your existing data, you may not reach the limit yet. You can still size up the model. That's basically a very, very important experience rate. So in that line, you can see there's something low-hanging fruit. If you have the right compute, if you know how to handle the actually quite daunting engineering issues, of training larger models, you actually will get a pretty, strong return in terms of model performance. Of course, for biologists, even for academia researchers, training larger model probably is not just a problem of machines. In fact, I ran into colleagues, which I say, how about I give you 200 machines? I don't think they know how to even use it because it requires you to really understand parallelization infrastructure, which I'm going to speak about in later part of the talk. In here, For example, we recently trained one of the largest protein model. In fact, not even with a traditional, dense and transformer architecture. It's a MOE, mixture of experts, which is one way to reduce computational costs because at a particular time, only a subset of the parameters are active and others are dormant. You actually have a way to distributedly train the model parameters and also invoke inference cost in a more economical way. But there are other tricks. For example, if your data have become really big, you need to put them on different GPU nodes, which is known as a data parallelism. If your model is really, really big, but they need to talk to each other in a coherent way, you have a smart model parallelism to make sure that they are consistent, right, and synchronous. And if your model has multiple layers, as we usually see in transformer and in LMs, you may even use a tensor parallelism or pipeline parallelism to really speed up the computing. So all this actually requires additional skill and knowledge beyond biology. And that's actually why, when you see modern science evolve, it's not the evolution only of the theory. Okay, many of the technical innovations, like the invention of the microscope, like the invention of many other like transistors and semiconductors, they are all very useful, in driving, the advancement of any discipline. I think in biology, we benefit a lot. In fact, biologists didn't build microscope or electron microscope or atomic microscope, but they use it. I think now it's time to use this particular microscope called transformers or large language models and so forth. So in here, this model is doing quite okay. We test them on over 300 different downstream tasks. Okay, there is a data set called the protein gene maintained by a researcher in Harvard, and which is becoming a very popular benchmark, to test protein language models. And you can see that we rank the performance, of our model against all these tasks. In some of the tasks, I think we do very well, and there are others which there are better performance. But overall, this particular model is doing decently, but there are room to improve. Along the same line, you can still play the size trick, on other data modality. We trained, a DNA model, foundation model, using a vanilla transformer architecture like this. Okay, but it is the biggest reported in the literature. And we test them over 30 downstream tasks in understanding genome contents, for example, making predictions about the function of the gene, making predictions about the activity and the program of the gene expression, and so on and so forth. And you can see that the AIDO model, overall is leading the scores, in I think 27 over 30 of the tasks. So getting the size bigger really gives you, a very, very immediate reward. Similarly, we did another model in RNA, also using a very, very standard vanilla transformer architecture. This time the model is a little bit smaller because the RNA data is smaller, but 1.6 billion is still the largest out there in the literature. And in this case, we get even a stronger bump. For example, we compare our model with a few other existing ones, indicated in here. And in many tasks, especially in predicting transcript abundances and so forth in different species, we improve the SOTA by a pretty significant margin. The only thing that is about sizing other models.
Can I interrupt you here and ask a question? So, so you mentioned the model, the scaling law, and then the model size, and also at the beginning you mentioned that the transformer may not be the right architecture for many of these problems.
Yeah.
So, what is your take on sort of the balance or the interplay between the innovation on the model architecture itself and also the size of the model?
Yeah, you will see that.
Okay.
Yeah.
Maybe,
it's a big space. That's why I say this paper is laying out 30 years worth of work, right? So we begin from the lowest hanging fruit size of the models, right? Because you want to make sure that you exhaust in there, just like what people do in GPT. They exhaust that first, and then you plug in one new innovation. In fact, you will see very soon that we have a few innovations to be plugged into already in our next iteration of the models. But in here, I just want to make sure that training a vanilla model with in fact here you already see a new architecture with depending on your real needs and also the data that you have. In here, for example, we are going beyond the experiment beyond sequence data. You can actually pre-train models on structured data. That wasn't done before, right? In the case of structure, protein structures are a continuous representation. It's very hard to search similar structures by matching the shapes and so forth. In here, we can pre-train a structure tokenizer, which is based on something called a VQ-VAE, okay, the vector quantization VAE model, which builds a discrete codebook of structures. Okay, and then you now represent every structure, using a code-down representation, and that gives you a lot of convenience in many downstream applications. Because, for example, you want to search homology between proteins. And nowadays, you actually could use this representation to give you very fast outcomes based on very novel definition of similarities, right? And they can actually even pull out the sequences or protein species which are very different in sequence, but similar in structure because of these structural representations. So we can actually compare this, to, for example, some of the state-of-the-art solutions such as, posit. We are actually doing, quite competitively with that already. And if you compare with one of the also well-known algorithm called DSM-3, we are, overwhelmingly, dominant in terms of performance. So this is just a small, kind of application of a pre-trained model using even the decoding phase, not even using the encoding phase, not even decoding, because if you decode, you can now reproduce the structure, which gives you a way of reverse, design the protein sequences. We also apply the same strategy to cell data. In this case, we did an exhaustive study about the gene expression data, to see what model size is correct. So we actually went through, a sequence of different sizes. And we start to see we are not reaching the bottom yet. Looks like when you increase the compute amount, okay, you actually could get a greater and greater performance when you enlarge the model sizes. But here in modeling protein or gene expression data, the architecture actually are actually facing challenges because we're talking about a transcriptome with maybe 20K entries, and that's a very long context. Okay, and so it's actually not typical for a transformer to handle that kind of context. Therefore, we are in the look about exploring new architectures. But with such a model, you can do actually already a lot of good things, such as embedding the cell in a informative representation, to visualize the clusters or to search for homologies and so forth. And here is a comparison of this model together with a few others, measured by different similarity metrics. So that's basically just the first part of the story. You have all the component models. Each of them can be individually, improved, with both engineering advance. Yeah, I want to really emphasize that people thought engineering is taken for granted. We should ask for new architecture and so forth. Well, I would say that first learn engineers. Okay, you as a biologist, as a team, now need to train yourself to use a new microscope first. And then we think about change the microscope. So this is about now, after you have all these neural models, what to do next. Because I said we want to have a system, not to have a bag of models. Therefore, all these different models have to talk to each other and integrate. And our stage 2 is really about connecting the dots between all different models. And there are a couple of technical approaches which allows you to execute that kind of cross-model communications and the integration dialogue. They are called evolve, align, drive, and cascade. And I will say a few words for each of them. What do I mean by evolved model? Well, basically, evolve means that you just continuously train the model after you finish the phase of pre-training with some better data or maybe some better representation of the data. For example, our sequence model are mainly based on one sequence at a time to train, right? But we know that there are multiple sequence alignment, which gives you homolog sequences as a cluster, right? You actually could turn that kind of a very specialized, MSA, grouped data, with the existing model architecture by playing with the positional encoding. here we tried an idea called high-dimensional precision encoding. That you have one dimension which encodes the token in their one sequence form, but the other dimension could give them an evolutionary position in the multiple sequence alignment. You could even imagine other dimensions to give them network positions and other positions, right? So in that case, you just use the older architecture, no change, but with a new way of tokenizing the information, you get more things out of it. And that's called evolve. It's the most computationally efficient and convenient approach to extend, but that gives you already a huge reward. We compare our model with, in fact, you can use that to now, make predictions about protein structures, right? And in that case, you not only have a sequence information, but also have evolutionary conditions, situations and informations, which is more like how AlphaFold 3 work. We know that AlphaFold 3, are way better than ESM, which is purely based on a protein language model. Why? Because it captures the evolution information. But in our case, we still have a pure protein language model, but with this evolutionary technique, we were able to be competitive, with AlphaFold 3, even though, we don't explicitly model multiple sequence alignment. In fact, in the regime where you have multiple sequence alignment, we do comparably to AlphaFold 3. But in regimes where, we are, there are no lot of homologous sequences, AlphaFold 3 actually will degrade very terribly because there is no support from evolution. But in here, we actually have a much more graceful degradation in terms of performance. The next approach is to align. In large language model, this has been now practiced a lot in building word models. You have the image domain, you have the text domain, and you want to basically align them so that they can start do crosstalk and do cross generation, right? And similarly, you can, for example, use a pre-trained protein language model to generate, tokens. And then you can also, get the similar structure tokens from a protein, model. And then you define, maybe a cross-entropy prediction function. Thank you. To really force them to align, based on some metric. They can, for example, you can use the protein token to predict the structure token, and so on and so forth. And that usually leads to a much improved, foundation model, which can gives you advantage, over structure predictions. And here is a comparison, of this model compared to the ESM3. You can see that, at this stage, we are already, stronger, in many of the metrics or data sets compared to ESM3. The third approach is to drive the model. Again, driven or driving, is yet another new way of connecting models. In this case, it's more like a sequential cascading effect. Imagine a task where you want to do protein reverse folding, inverse folding, meaning that you have the structure, you want to generate the sequence. And what the structure requires a protein structure pretrained model to generate the tokenizations, representations. And then they need to be fitted into a protein language model to produce, the sequence outcomes. And that's what I mean by driving. You can use a adapter, which builds on some kind of a cross-attention mechanisms to try to connect the protein representation, structural representation with the sequence representation so that you can finish this pipeline about the driving generations or conditional generations based on different data modalities. And yet another example in driving, now we have, say, the cell model I just mentioned, how to drive it to have an interesting effect. Well, maybe one thing you want to know is to predict the outcome of a perturbation. I knock out, I crisp out a gene, and then what happens? Well, that signal of perturbation needs to be properly encoded using maybe a perturb model, right? And then it's usually based on an interactome, which tells you how genes interact with each other or correlate with each other, and then that information can be used to drive a pre-trained cell model to predict the rest of the transcriptomes, the gene expressions. We actually compare this with a gear model, some standard benchmark. You can see a pretty strong performance improvement because here you are now utilizing differently pre-trained models, which can be connected through an intricate kind of co-attention mechanisms. Last but not least is maybe the most exciting and valuable kind of a way of using all these different models, which is to really tie them together in a cascading way, because biological system is a nested hierarchy. We all know that, right? So we started from an organism, breaking down into organs, tissues, and cells, different cell types, and so forth. In a sense, the representations coming from the bottom can actually be utilized to encode the information of the next layer. For example, if you want to make a prediction of a protein-protein interaction, for example, how people do that? Right now, most of the machine learning approach is to build a network model, okay, and therefore use the graph to predict basically, whether some pairs are interacting, and you can turn them into a graph neural nets. Same thing, it's to really capture topological information in the graph. But remember, every node in that PPI is actually a protein. Protein has structure. Protein has sequences. How to actually incorporate the structure and the sequence information into a prediction? Well, in this case, you can now use a representation, learned from the protein language model or protein structure model, and then directly use that embedding as a representation of the node. And then you finish the cascading of the information above to a higher level prediction. Maybe more obviously is the an ongoing project where we want to now use the cell model to make stronger predictions of cellular behaviors such as knockouts and so forth. How to represent a gene? Many of you probably worked on the current cell foundation models published out there. What is a cell? What is a gene? It's an index. It's number 2. Or it's MSK and so forth. It's just a name, right? And then you somehow encode it. And you encode a stupid thing, which is a number, which doesn't have any biological meanings. Right? But if you have a sequence model, if you have expression models, they actually gives you functional representations of the embedding of the entity. Therefore, your gene is represented by a meaningful embedding that carries the sequence information, structure information, and functional information. And then you use them as the input of the cell model, which will be predicting the cell behaviors. It's a much more logical way of doing the biological simulation, but it requires 2 or 3 or more different models to be at hand, and then they do talk to each other. In fact, you can even evolve it further. Once you have this in hand, you can go temporal spatial dimension to evolve and cascade the model, right? For example, what if you want to predict the behavior of the cell, also conditioning on its neighborhood? Because the cell is not a singleton, right? I have a whole bunch of cells next to me, and they would influence also my effect, right? And how to represent that? Well, you have the cell representations, embeddings, which also can be a vector input into your predictive machines of these models. So hopefully after seeing this, you can kind of see how bizarre right now that biology is done. We're assuming a cell is a singleton. We're assuming a gene is a number. We assume basically, the gene expression is a graph, and we use that to make predictions. I have to say that none of these predictions probably make sense at the end of the day, because you give away 90%, maybe even more of the information actually existing in biology, just because you don't have the right tool computationally and also algorithmically to consume all this. That's why I think foundation models, especially a system of foundation models at multiple scale, is essential. It's not an option. It's essential to make a sensible predictions. Okay, so I'm going to wrap up here. So here is a summary of the technical results I just presented. They will be published in the forthcoming New York conference workshop of drug design and others. Here are the titles of the paper. And I think they should be online already. If not, next week should be there. And also together with the position paper, you hopefully can see the bigger picture about what we are doing. And all these papers, and the results are very comprehensively tested, because we meant to create a community which can share the results. So we actually paid a lot of engineering depth in building the tool stack and also the data set which are now exceeding 300 benchmarks, for all these tasks, and we are going to announce it for public use. So these are, you probably see that we are still missing a few models, right? I just presented maybe 5 to 6 models. We are working on, for example, a new version of the interacton. We are now also working on a tissue model and a phenotype model, which obviously will be using higher level informations. And you see a logic of why we do this later, because all these models requires the entities in the model to be represented by a lower model, a representation, and that's what we built first, right? Of course, all these models will keep iterating by increasing the size. I think scaling law is still not fully exploited. The difficulty is, do we have enough money to get enough machine, or do we have the engineering power to really, operate on large machines? And we want to overcome that first, because these are the lower hanging fruits. Designing the new model architecture will be harder. We'll be doing this slowly. And at stage 3, what if when all these are done? Are we done? Well, not yet. Imagine you buy a car. Your first 2 or 3 months will be just driving and fitting the car and just harmonize the whole thing. It's just like an orchestra. You bring all these great soloists. They need to rehearse a couple of times before they really can play together, right? So that means all these models eventually will put together and gets harmonized by, for example, co-training, co-fine-tuning, solving multiple tasks together. So all these actually requires additional technical work that we actually don't even know how to do it, because here we're talking about embedding across the layers and also track feedbacks and the gradient flow across the level, and nothing like this has been done yet, not even in LM world, in GPTs. So there are a lot of interesting area that can even goes back to the core of computing and AI beyond biology. So that's why I think this project is super, super exciting. And of course, we are biologists. We don't want to only make prediction and just SOTAs to utility. We do want to advance the science. So we imagine that these multi-scale foundation models eventually become a critical link in this full circle where we do experimentation and validation in a guided way. I think right now, when wet labs are producing data, it's not very purposeful other than a very localized experimental design and for a specific disease. But if you have the bigger picture in place, you know where you need more data and where you need more validation. I think we can make this whole ecosystem much more productive and efficient. And I also want to argue that doing science in this way is safer. People always ask, is a biofoundation model safe or not? Well, I ask, is your lab safe or not? You do your stuff in the test tube, and you need to have all sorts of protections, on that. We do that in computer, you can shut it down and nothing leaks. Okay, and if you get the information, you still need to go to test tube, to synthesize them into a real material. Therefore, there are additional guardrails and protections and checkpoints. So I would argue that doing the biology in a silico, simulationally, is way safer than tampering with your materials and risking a virus going out of your lab and so forth. So just a very quick summary, maybe for a different audience that can spend more time, there are a lot of technical challenges, all the way starting from designing new transformer architectures or maybe even novel architectures beyond transformers, setting up different supervision tasks, such as invoking new loss functions. Mass language modeling or causal language modeling may not be the right thing to do, in biological world when they want to do constructive learning or many other, multi-objective machine reinforcement learning, because when you design, you may get a feedback, how to incorporate that. So there are a lot of interesting things going on, to be developed. And then last but not least is high-performance computing needs to be rebuilt or refurbished, or not repolished, for this particular application. Here, actually, because my own interest in HPC, I do want to say a few more words to raise awareness and attention and appreciations. What we did, what make us possible to do all what I just presented, is exactly because my lab used to be a HPC lab. Okay, we actually invented and advanced many of the parallel computing structure and technology in the past, we actually use them very, very aggressively, and we do more. So for the task stack over there, you see, different type of task, ML task, but also Gen task, and also different benchmarks. Without a standardized platform and toolbox, it is very hard to form a community to do them in a comparable way, right? So we are trying to now provide such a toolboxes, but also even for the training framework, there are also a lot of room to improve. For example, there are tools for auto ML, tuning the parameters in a more efficient way. And certain quantification is also not available in any other biological work, right? And then all this explanation, and the causal inferences within the model is not feasible at this point. So we want to actually include some of these toolboxes, in our offering, so that we have a very limited bandwidth, but the community come together could utilize all these different resources. And here, of course, is another kind of team sitting at the bottom to manage all the machines and the infrastructure. And there are a lot of ingenuity and creativity devoted to GPU clouds, data management, storage, and code. And here just give you a sense about the scale of that work, right? So the work that you see now, was done on a cluster of 500 GPUs. And 500 GPUs is actually pretty expensive and very, very difficult to manage. And because it's expensive, we don't want to have a single minute that it is idle. Therefore, scheduling and programming becomes really, really hard. And we also have a team standing by 24/7 to make sure that when any node goes down, you need to have a way to rescue it. So that's basically a kind of a rough picture about what it takes to work on a project of this magnitude. And here is the offering that we're going to announce next month, actually this month, next 2 weeks, a software package called Model Jian, where you can download the pre-trained models. You can apply all the fine-tuned downstream task scripts, and also you can use the data which are standardized and formatted for your experimentation. Of course, we also wish people would contribute to make this repository even larger. Now, maybe I should just mention a few words about the applications. Now, obviously, if you have a simulator biology at all level, there is a huge space for applications. For example, you can start from molecular engineering, right? Talking about protein design and other functional generations. And here, there is opportunity to form a feedback loop using the reinforcement learning to actually further fine-tune and train the model. There are also a potential to do cell engineering going one level up to really try out, different ways of perturbing, a sick cell, to reverse them to the right course, or just, let them to die, faster, in various different ways. But again, you don't have to do it in a test tube. Hopefully you can do it here before you try the last few most plausible hypotheses in your test tube. We can also do phenotypic engineering. Right now, one of the projects is to really leverage on the ability of a genome foundation model to encode or embed arbitrary polymorphic genes. Remember that in our current GWAS study, you are relying on the statistical power of the SNP to pinpoint diseases. And the SNPs are all over the place inside a particular gene. And therefore, the statistical power is really weak because on every particular location, you don't have too many variants, right? Variants. But if you have an embedding, of the whole gene, or maybe of the whole operon, or maybe the regulatory sequences, of these polymorphic sequences, you have a continuous spectrum, of the gene instances in the whole space of embedding. And in that space, when you do association mapping of complex phenotypes, which can also be encoded in the encoder, in the later representations, you actually are having a new paradigm of doing association studies. So in that way, I think phenotypic engineering can be also very promising. Last but not least, there's an interesting twist about how to engage and play with these models. I think one of the secret sauce behind the GPT is that it is so playable by anyone. You don't have to be a trained scientist. You can now program maybe even through natural language because there's an interface of natural language. All these protein models don't have a natural language interface. You cannot say, hey, AIDO model,
build me a protein with a melting temperature of 50 degrees. You cannot say that because they don't understand it. But what if we put a natural language model, aligned with our biofoundation models, so that you can start talking to such a system, the AIDO system, within natural language. That's actually quite doable. In fact, people do that already in other domains. It's really about finding, an aggressive way of aligning data from different modalities. But I think, I'm very hopeful because the literature of biomedicine and the molecular biology is so rich. Many of the genes, have a proper natural language description of their functions and activity and so forth. So we are working on such an interface as our last applications. So to sum up, I want to reiterate that every human health problem is actually a biological problem. Therefore, understanding the biology at all degree and all level is really, the key to unlock many of the opportunities to solve, healthcare problems, medical problems, pharmaceutical problems in an economical and a more effective way. And I think AI is right there to provide the tool beyond, a certain slice of spectrum, unlike what we did in the traditional classical approach. And again, to revisit this kind of a dichotomy, what we do, we present it. I want to make a disclaimer. It's not first principle. Okay, I'm not trying to pretend that I'm, discovering the secret of biology, because that's never the intention. The intention is to predict biology and program biology and simulate biology, with or without knowing the biology. Okay, and that's why you see AlphaFold, start to be very, very functional and performant. This is different from, here you use a Newton's gravitational law to predict the planet orbit. You use a periodic table to predict the property of the chemical compound. That's great if we can do it, but I don't want to wait until they are available before we want to carry out useful functionalities. So that's the end of the presentation. We are hoping that this system, so-called AIDO, the AI-Driven Digital Organism, can be a new paradigm for holistic life science, because they are holistic. Okay, the model penetrates all data modality and the scale, and the problems you ask basically, hopefully, can be covered in one of the particular spectrum or band of the spectrum, with all the rest of the spectrum supporting the site information and the context, so that you solve every biological problem in a contextualized way and also in a holistically simulational way. I think with that, I'm done with the presentation.
Questions from the audience? At one point, there are like over 40 people online. So
now what?
I wonder if you can ask them if they have any.
Yeah. For the people who are online, feel free to ask questions. I think I'm muted. You can just speak out. Questions here? Like.
So that's great. I think it's a lot of work and impressive. So can you talk a little bit more about simulation versus using model to predict? As a simulation, you need to have more understanding of the interaction of the molecule even at the atom level.
Mm-hmm. Technically, simulation and the prediction are very similar behaviors computationally. But I think by prediction is a one-shot thing. You basically ask a query, you have a sequence, you ask what the function is. That's basic prediction. But a simulation is more like you can imagine, for example, a counterfactual experiment. You provide multiple scenarios, in perturbing the cells, which hasn't been perturbed yet. And you want the cell to simulate, the system simulate what happened, for the entire cell and so forth, right? So I would say the simulation also includes a feedback, which actually could, have a sequence of queries and maybe, feedbacks about the consequences and the scores and so forth. That's, I mean, I don't know why in the world they define generative model, because a generative model requires a decoder, which can be preconditioned, on various different things, right? But for prediction, you don't need a decoder. All you need is an encoder with a latent representations, and then you can use that representation for whatever thing you do next, build a classifier and so forth. So technically, yeah, there is a different treatment about these two things. I think in this case, I showed the pre-trained model as a template, to provide you the right representations. But then we also offered this tool set people can fine tune, and also a tool to simulate, in user specific fashions.
What does it take on like, because you mentioned this first principle and then empirical evidence from like in the data-driven manner.
Yeah.
But there is I think there is a challenge in biology right now is even though there are a lot of data, but we may not have the right types of data for many of these problems. On the other hand, we do have a lot of knowledge, right, from literature, and you mentioned a little bit, from biomedical literature. And there's also recent work of people showing that, if you want to represent a gene, right, let's say that just the embedding of the gene, a GPT-4 actually can generate pretty informative embeddings of the gene compared to what you can learn from massive amount of single cell RNA-seq data from gene expression. And what's your take on sort of these like there are different data-driven approach, but also there's this knowledge-based like existing biomedical data that knowledge and literature you can apparently leverage, and also like what type of data that we really need to build a model like what you have envisioned.
I think the GPT results are cherry picking. I can easily say ask another question and take another answer, but it exists in my data. I think that's not even a meaningful thing. Remember, knowledge, at least biological knowledge, I believe come from data. Even our textbook knowledges, if you go back to their origin, it's always a paper that published AGL or experiment. They may not be the data that we currently use, but there are data out there. Yeah, I would say we actually have more data that can be used at this point. That's why methodology has a room to further advance. But also, it's a matter of technical kind of trajectory. Just like I think right now we are in maybe the GPT-1 phase, of biology. At that time, you have all the NLP people having all these different algorithms still able to beat a GPT. One that one was not even impressive results. On many things like a parse a sentence and so forth. And they will say, I have a knowledge of grammar, I have a knowledge of history and so forth. They can do it. But look, after 3 years or 5 years, when GPT gets really big, okay, and basically, they become a superset of your knowledge because your knowledge comes from. So the debate of Kantism and humanism is a philosophical one. I put it only here just to give people maybe an alternative space to think, because we were also programmed and predisposed to a first principle thinking. We thought we need first principle to do everything, but I tell you that you don't. Okay, you go to a hospital, you drive, you do many things based on a lot of instincts, based on a lot of experiences. You must remember everything. In fact, if you are a musician, you play, and most of the time, you don't necessarily play through a real-time understanding. You must remember everything already. So at least this is an alternative. I'm not saying this is better, but it is alternative. Now, when we look at the first principle, when it fails to give you the answer you need, then what do you do? I think that's the most important thing.
Okay.
Right. I'm out of time or what?
There's a defense.
Okay, sure.
Sorry.
More questions? Perhaps?
Audience.
Thank you very much. Yeah.
Thank you all from the audience joining us. Yeah.
Okay.